Hey all, so today we're going to be looking at A2.1 and A2.2, which is the origin of cells. And so the questions we should be asking ourselves are what hypothesis could account for the origin of life, and what intermediate stages could there have been between non-living matter and the first cells? So this contraption set up here was put together by Urey and Miller. And what they were testing here was if we could have these gases, ammonia and methane and hydrogen gas, in a boiling water, so there's water vapor there too, and electricity, could we produce the earliest precursors to life, i.e. amino acids? Because our prebiotic atmosphere had methane from volcanoes, tiny amounts of oxygen, and CO2. There was no ozone layer, so there was a lot more UV radiation. And we want to know, could it have generated carbon compounds if there was lightning? We're still unsure what the temperature or the pH of the oceans were at that time. But what Urey and Miller showed in their experiment was that carbon compounds potentially could have arisen from Earth's earliest atmosphere. So Louis Pasteur was a guy who falsified this concept of spontaneous generation. And so living things are self-sustaining and in an ordered state. They use energy and they can reproduce. And cells are the smallest unit of life. Some cells are dead, like hair cells, but others are going to reproduce themselves. And so what Louis Pasteur falsified was this idea of spontaneous generation, where living things initially a few hundred years ago were thought to arise from non-living stuff. So that would just be like, poof, a rat is on your counter and it came from nowhere and it's just magically there. So the way that Louis Pasteur falsified this was he had some like broth, which is full of nutrients. So like think chicken soup, and he boiled it. And then he had these swan-necked flasks here. And with one of the boiled ones, he didn't open the flask. So this neck wasn't broken. And then the bacteria couldn't get into the broth. But in the other one, he boiled it and then broke the neck. And so the bacteria could get in. And shockingly, the bacteria grew in the broth that was open to the air, but did not grow in the broth that was not open to the air. So he falsified the idea of spontaneous generation, but then that raises the question, where did the first cells come from? And they would have arisen hundreds of millions of years ago. But in order to do that, they would have needed some kind of catalytic reactions. They would have needed to be able to catalyze their own reactions in their own bodies. They would have needed to self-assemble, so like building amino acids into proteins. They would have needed compartments, and they would have needed to be able to replicate themselves. So if we're thinking about compartments, phospholipids can make vesicles because the head is attracted and the tail repels water. So you could, in theory, have compartmentalization happening because spontaneously the phospholipids were lining up in such a way that in the carbon soup that they were making these little compartments, and that would have allowed for a membrane to slowly form. RNA can catalyze its own processes, i.e. the joining of amino acids in the ribosome. So this may have been the first genetic material. That's the genetic material in viruses. Also, RNA has a much higher mutation rate than DNA, so it could allow for lots of different changes to be happening, especially in a higher UV environment. And then what we are seeing today is that we all use the same genetic code, and that genetic code has the same 64 codons, and we all come from a common ancestor. And so we refer to that common ancestor as LUCA. LUCA stands for last universal common ancestor. And so there would have been some OG cell or protocell that formed, and from that, through different mutations, the three different kinds of organism systems, eukaryotes, archaea, and bacteria would have formed. So that evolutionary timeline, if we look at Australia, we can see that these stromatolites, which are these hydrothermal vents here, show life from earlier than 3.42 billion years ago. Earth was formed about 4.5 billion years ago. So the isotope ratios of carbon-13 to carbon-12 is low in living organisms. And this is seen in some rocks in Greenland, and those rocks are 3.7 billion years old. So that's suggesting that those rocks have some organic matter that was once living in those rocks. LUCA genes have been identified from shared bacteria and archaea genes. So there's 355 protein families that were likely in LUCA's genome. And that the types of genes that these have and the protein families that these would have related to the genes suggests that LUCA lived in a high hydrogen, high carbon dioxide, and iron environment. So that might have been in hydrothermal vents that were quite hot, between 60 and 90 degrees Celsius, that were alkaline, meaning they were quite basic, and they just had lots of free hydrogen, methane, ammonia, iron, and sulfur floating around nearby. And that could have allowed LUCA to create its own energy. It would have needed some kind of some genes for anaerobic respiration so that it could fix carbon dioxide and nitrogen from the environment. So endosymbiosis is something that is going on to create eukaryotes from prokaryotic organisms. And so this would be a beneficial symbiotic relationship where a type of prokaryotic organism was absorbed into another, and then they lived together. And that absorbed organism provided food and energy while the engulfer provided protection. And so initially, this would have been like the proto-mitochondria. So it was engulfed, and endosymbiosis means engulfing. And so this prokaryotic host cell engulfs the small mitochondria, and they slowly changed over time so that they continued to work together. And there's evidence. And so that was step one. And then step two would have been the engulfing of chloroplasts. And so chloroplasts and mitochondria are quite unique organelles in that they have their own separate DNA. They have a double membrane, which is coming from their OG membrane. So they would have had their own outside membrane initially. And then the engulfer also would have put a membrane around them. So they have two membranes. They have different ribosomes than their host cells. So they have 7DS ribosomes instead of ADS. And they need to reproduce from pre-existing mitochondria or chloroplasts. So they're acting like cells, separate cells. But this would have allowed cells to become bigger because it would have allowed cells to become more complex, also allow them to become bigger because they would have been provided with more energy, and then also allow them to differentiate. So when we're thinking about cell differentiation, it is more efficient because it allows cells to be specialized in their job. So in any given cell, only about 4,000 genes are active. And in different cells, different genes are activated. So my eye cells don't work the same way as my white blood cells. And that's, even though they all contain the same amount of DNA, only about 4,000 genes are active. So multicellular organisms have arisen multiple times independently. And multicellular organisms tend to live longer, are larger, and they can be more complex. So they're going to live longer because if I burn my skin and some of the cells die, my whole body does not die. And they can be more complex because they can differentiate their cells to do specific jobs. Most life, however, on Earth is single-celled, which suggests that overall it has an advantage on complex life like humans. So to summarize, early atmospheric conditions could have led to formation of materials needed for cell formation. Though at some point they did, generally cells don't spontaneously generate, they come from somewhere. And this spontaneous generation process would have been a very, very slow sequence of steps. Cells likely evolved near hydrothermal vents, and there is evidence for a common ancestor because we all use the same codons, the ribosomes, the enzymes, and the DNA, RNA for synthesis. So our processes are all identical, which means we are not converging evolutionarily. We came from the same OG space, like we have a common ancestor. And then endosymbiosis allowed for the formation of eukaryotes from prokaryotes. So that was sucking up a mitochondria, which wasn't at the time a mitochondria, and then secondly, a chloroplast, and mitochondria gave you energy from sugar, and chloroplasts gave you energy from sunlight. And so if you're providing a home and that you're willing to share your home and protection in exchange for energy, then you are going to be more successful as a cell. All right, that's it for this video. Thank you for watching, and more soon.